Hello, everyone, and welcome to today's webinar, Single Cell Antibody Discovery Using Islands of Automation. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit them at beckman.com. Now, we encourage you to participate today by submitting any questions you might have during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. I'd like to now welcome our speaker, Paul Anderson, the Director of Biologics Discovery Automation at Lilly Biotechnology Center. Paul, welcome. You may now begin your presentation.
My name is Paul Anderson, and I'm proud to present the work that we've been doing at Lilly for the last several years in automating a single-cell process used for biologics discovery, specifically antibody discovery. I joined Lilly 2 years ago, so a lot of the work I'm sharing today is the work of a fairly large group of people, and some of the people that helped make this presentation and had a major role in developing these systems are my co-authors, Mike Cohen, Melissa Crisp, and Tom Kujak. So I just want to thank them for the opportunity to share their work and the group's work today with everybody. So I'm going to dive into talk about the process that we're automating here. We have several antibody discovery platforms at Lilly, and the one specifically I'm going to touch on today is immunization-based platform using single B cells and going through a series of automated platforms to generate lead molecules. We have several other platforms, as I mentioned, that are orthogonal to this approach that we use for antibody discovery as well. So this is the main process I'm going to be talking about is this single B cell application. But keep in mind, a lot of these systems are used in different aspects of other biologics discovery efforts. And so this presentation is really just honing in on this application here. So we have several platforms I'm going to discuss throughout this process, and these are really all tied together with a LIMS infrastructure. So we are working on collecting information from each of these platforms in the form of data as well as sample tracking. And so really one of the overarching themes here is the software side of the process, where we're capturing a significant amount of data, whether it be purification amounts from DNA or purified material amounts from expressed antibodies, data and associated with titers, and a significant amount of screening results toward the end of the process. And what we're trying to hand over is really a data package so our project teams can make informed decisions and triage down to lead molecules as fast as possible. So our process, this particular process starts with the immunization-based strategy, as I mentioned. So that could start with mice or a variety of different host organisms or host species. In this example, we're using a mouse model, and so we would immunize that mouse with the antigen of interest. And then once we have sufficient serum titer, we would isolate the B cells and use a back sorter to dispense antigen binding B cells into 96 well plates. And so we typically start with roughly 20 96 well plates. So the process I'm going to talk about here, usually it's about 20 96 well plates, so about 2,000 single B cells will go into this process as this first pass. There's several projects where we go through multiple passes, several immunization strategies, and so they grow significantly larger in size. So the first system I'll talk about is what we call the fill and barcode system we developed with Beckman. And this was essentially a automated liquid handler with a series of other third-party devices on there, such as a Cytomat, a labeler, a sealer. We added a balance recently. And really this system is designed to add reagents to plates, barcode or label those plates, seal them up to feed several platforms throughout our process. With the the case of where we're sorting cells into plates, we typically are using, those plates are created on here with cell sort buffer and sealed up and stored in a chilled manner until we're ready to sort cells into them. Then once we sort cells into these 96 well plates, they come back onto this platform when our scientists are ready to process them, and then they are consolidated into 3D 4-well PCR plates. And so the next system I'm going to talk about, where a lot of our molecular biology takes place, this system feeds that as well, adding different PCR master mixes to a variety of 3D 4 PCR plates. And so the plates come off of here. They've been consolidated into 3D 4-well plates, and they would come to what we call a molecular cloning system, also developed with Beckman. And so on this system, I'm going to touch on some of the usual suspects of instruments that you'll see on several of the systems coming up. And so that would be a plate lock and X-peel, and those are for sealing and peeling of plates, as well as a V-spin, a centrifuge. And so that is typically to eliminate bubbles and pull samples to the bottom of a well. And there's several cytomats on our system. So we have an ambient cytomat on this system, as well as a chilled cytomat. So as we go through the presentation, I'm really going to try to touch on what makes each of these systems unique in their own right. So the molecular biology and the molecular cloning system, the kind of special sauce on this guy is the bank of automated thermocyclers that you can see in the top. So there's a stack, 3 stacks of 4 thermocyclers, so a bank of 12 automated thermocyclers on the system. And this is what we use to carry out a series of PCR reactions on here, where we start with the single B cell sorted into each plate. We split it to heavy light chain and assemble it into our vector of interest. So the output of this system is going to be heavy and light, a set of heavy chain plates and a set of light chain plates assembled with the antibody gene of interest. The first QC step in our process incorporates, on the top left here, you see an ATI fragment analyzer. So we cherry pick samples after the second round of PCR, and this really, we run these on the fragment analyzer, and this lets us get some insight as to, are we chopping out the fragment of interest and isn't it the right size? And so this is, usually we have a fairly high success rate of this, but if there's anything going wrong with our master mixes or anything went south in the sorting side of things, this is where we would typically catch problems. And so as you come off of this system, we have our heavy and light chain DNA. We go into a semi-automated E. coli transformation step. And so we, in a 3D 4-well manner, do a transformation into competent cells, let that sit for a bit, and then transfer those into 96 deep well plates with LB broth. And those will sit in shaking incubators overnight. And after that incubation overnight, we pellet the cells, we centrifuge and pellet the cells and decant them and seal them and store them at -20 degrees until we're ready to do our plasma purification. And so the next part is we built a recent system to do our plasma-based purification. And we are using the 96 turbo kit from QIAGEN. And so what you see here on the top left is the E. coli pellet block. We have to dispense a few different reagents to resuspend, lyse, and neutralize the E. coli. And then we transfer that material into a lysate clearance filter plate, and samples go through that to clean up the garbage that's in those samples, that's kind of snotty, viscous material. And it gets eluted into a DNA binding filter plate. You pull samples through that filter plate, DNA binds to the membrane, do a couple rounds of washing, and then elution to purify to do your plasma purification. And so as we approach automating this, our manual workflow involves vacuum manifolds, and after this last wash step, we would do a centrifugation step to really give a hard spin to these filter plates to get rid of any residual wash buffer before we do our elution. Automated vacuum manifolds are quite common, but there are significant drawbacks when you have samples that could be, if you have certain wells that don't have samples, the vacuum is going to pull through the path of least resistance. If you have other wells where are slow or clogged a bit, then those tend to not get pulled through. Once you have wells around them that get pulled through the vacuum, the ones that are clogged or running slower tend to be more and more difficult to get to be able to pull through the filter plates. And so we, in talking with Beckman, we had explored some other options with how to proceed with handling filter plate-based purifications. And so we had the idea to explore positive pressure devices. So we had this discussion with Beckman. They talked about some positive pressure devices, and then we started looking into it more and found several options on the market. And so we brought a few different devices in that I'll talk about on the next slide. But to just give you a little bit of a background on what these devices are and how they differentiate themselves from vacuum and centrifugation. The way they work is pulling in house air. It could be a gas, nitrogen, or different type of gas into a pressure reservoir. So you have a high pressure side on the reservoir, and it uses a flow restrictor, and this is essentially software controlled to open up small holes and let some of that gas or air through. And so the nice thing about this, you're essentially pushing samples through a filter plate. And whether you have an empty well or a well that's completely clogged, since you have a large pressure differential across the restrictor, you have the same amount of pressure being applied to every well. So whether it's empty sample or a clogged sample, you don't have those wells impacting the nearby wells or adjacent wells. And so it's a very nice, consistent flow rate across the plate, which is highly desirable. And the other thing is, essentially, to get into these positive pressure devices, you're stacking and unstacking labware. And so that's a big benefit as well, because that's a fairly simple mechanism to automate. So while vacuum is very inexpensive and easy to automate, it has some quality and reliability issues that we would be concerned about. And so the centrifugation method, you do get even flow across all samples, but is a bit more challenging to automate stacking and unstacking and the waste associated with plates going in and out of the centrifuge. And so that was a bit more of a red flag for the centrifuge-based method of handling filter plates. Whereas positive pressure, it's a bit more expensive, significantly more expensive than vacuum, kind of on par with automated centrifuges. But it's very fast, easily automated, and the flow control is very good. And so we opted to explore this route a bit further. What we did find in our due diligence is that this is essentially this is very new for most vendors. And so the ones that we reached out to, they were just starting to automate these types of devices. And so we brought in 3 devices from 3 different vendors. And what I'm showing you on the top right here is in blue our heavy chain DNA and in red our light chain DNA. And so we have control samples which would be done manually using our vacuum and centrifugation workflow. And then all 3 devices performed well in our hands. And so you can see there's a bit of a bump in DNA concentration with the heavy chain and a significant increase in DNA concentration with the light chain plates. This was I would throw out the disclaimer that this was a fairly quick and dirty experiment using these 3 different devices. Not a lot of optimization done to really dial in flow rates and dial in other conditions related to these devices. So as a first pass in some early data, we were very pleased and encouraged to explore this technology further. What we did as well, I mentioned in the first system, we have automated balance on there, and you'll see that throughout the systems I'm going to talk about. We weighed the filter plates before and after each of the liquid handling steps. And so to explain the higher concentration on here, what we think is happening is after you do your last wash step, when you are pulling that through a vacuum and then going to a centrifuge to spin out as much residual wash buffer as possible, there was, based on gravimetric results, more wash buffer left on the filter plate than when we were using the positive pressure devices. So we believe that we were effectively eliminating, pushing through more wash buffer, more residual wash buffer on the positive pressure devices. And then when you go to your elution, you've reduced that residual wash buffer more effectively on the positive pressure devices. And so our elution had a higher concentration of DNA. So that's really what we believe to be the main cause of the increase in concentration. And so we did look at the quality of DNA. We ran it on gels, and I'm not showing that data, but it was right on par with the positive pressure device in our normal workflow. And so we believe the quality of the DNA was good. And in the bottom right figure, I'm showing expression titers. And so what really matters at the end of the day for us is how the yields look. When we take the antibodies, the DNA that's purified on these, and we go into transfection and look at what the antibody yields are. And so black bars are a couple replicate plates, and then red, purple, and blue are the 3 different vendors, positive pressure devices. And there is some variability in here, but by and large, we have run-to-run and plate-to-plate variability that's within this degree on our transfection system. And so we believe this, the DNA produced from these positive pressure devices was right in line with what we were looking for. And so it was nice to see those results. And so we opted to go ahead and work with some companies and ended up working with Beckman to design a DNA purification system. And so the first system that was proposed was a Biomech i7 with 2 positive pressure devices, and they partner with Amplius for these positive pressure devices mounted off the side of a Biomech i7. And then we have, again, plate lock, XPO, multi-flow for the reagent dispenses, and some bioshakes on the system as well. And we have a reader on here. So as we purify DNA, we can go take an aliquot and measure it and determine what the purified yields are. And so what became apparent as we first designed this system and looked at what it would take to run 40 96-well mini preps on here. What I mentioned earlier was we started this process with 20 96-well plates of sorted cells, and those get split into heavy and light chain plates. And so that's where the 40 96-well plates are coming onto the system. So when you look at the plates that are coming in, the tip boxes required to transfer samples from the E. coli pellet deep well plate onto the filter plates themselves, elution plates, and tip boxes to transfer to make that aliquot of eluted material. There's a lot of labware you can see associated with this process, as well as looking at the time in the models with Beckman was 11.5 or so hours just for the DNA purification side of things. And so this led us to start exploring another bottleneck on here, which is all the labware that we have to have on this system. And so what we took a look at was the thermo skyline that had come out right around the time, or just before the time we were starting to design this system. And so this can be thought of like 14 PEZ dispensers. And so without having shelves and you don't have a random access like the standard Cytomats, you essentially can stack up filter plates, stack up your tip boxes, and really have a high density storage device on here. The other thing I mentioned is we have a reader on here, and we are able to measure how much DNA we've made on here. Well, the next step is to normalize and mix your heavy and light chain DNA, and I'll talk about that in a little later in the talk. But since we have the information on here and we have a liquid handler on here, we have all of the tools in place to be able to do that mixing. And so we modeled that up as well. And so that's where you see additional labware on the right. So we have another set of tip boxes. We have the mixed DNA plates, which should be 96 deep well plates. And then we make an archive copy, which would be the 384 well copy with a small amount of DNA. And that's kind of like our insurance policy if anything goes wrong with the original plates that we've made. And so when we model the DNA purification with the mixing, we're looking at roughly a 20-hour workday or 20-hour automated process to go from E. coli pellets all the way to DNA that's mixed and ready to be transfected. And so that was it doesn't leave a lot of time if you're going to do this in subsequent days. And a lot of this is going to be running at night. And so what we wanted to look at, we're also exploring a couple new devices on here, the positive pressure devices, as well as the thermocytomat skyline. And so to de-risk ourselves a little bit, we worked with Beckman, and they had a nice proposal on upsizing the system a little bit. So what we have here is what the final system layout, and it's another i7 with 2 more positive pressure devices. And what you see are the skylines are mounted directly underneath the Biomech i7s. And with all the labware that we're moving around, the robot movements start to become one of the bottlenecks on here. So by mounting these skylines directly underneath and having the Biomech pull tip boxes and filter plates directly out of the skyline and move them onto the deck, we're minimizing transport times and speeding up the system significantly. And then having the redundancy of essentially a mirrored liquid handlers, mirrored positive pressure device and purification systems on here gives us a bit of an insurance policy as we, anytime you're automating new types of instruments, whether it be the thermocyclers or the fragment analyzer, there's always unforeseen things after you go through a significant number of samples over a couple years. You start to find what things work and what things are a little bit not as reliable or robust. So by having some redundancy on here, we really are happy that we have some insurance policy to keep the system running at all time. And so we've put a balance on here as well. The balances are throughout the system you'll see are really to do some automated verification. So as we tell our liquid handlers to dispense a certain volume or to transfer a certain volume, we were weighing most of the plates through all our systems before and after each of these steps. And this is our QC step to make sure our liquid handlers are holding up as expected. And so we have the positive pressures in place. We have high density storage using the new skylines. And so with the new model for this system, with some parallel processing between the two wings of the system, we can get the purification down to a little over 7 hours and then mixing in about 4 hours or so. So this is pretty reasonable for the number of plates we're putting through the system, a little over 11 hours for a full purification and mixture. And so from there, we go to the protein expression system that was developed a few years back with Fujifilm, or Waco at the time, and now Fujifilm. And so we take those 96-well plates with normalized and mixed DNA, we put them onto this platform. We add our transfection reagents and cells, and then go into these Cytomat 2C tower shakers. And so these are shaking incubators. And some of the nice features of this system are we have 4 Cytomats that each have a 14-plate capacity, so 56 total plates can run at a time. Again, when we developed this system, these tower shakers were early days, and so by having a bank of 4 of them, that's been a nice insurance policy where we've had issues that pop up from time to time. We're able to continue running. Typical runs are in the 20 to 40 plate range, so we're not using all 4 at a time for most runs, and so we're not using all 56 positions or all 4 Cytomats at a time, and so we've been able to get by and keep our process moving forward by changing the configuration of these on the fly and using as many Cytomats as needed for a given day. When we go through our process, right now I'm talking about a 96-well process, and we're doing close to a little under 1 mil expressions on this system. We eventually get to a point where we triage these samples down and people need more material, and we'll purify samples. And so what runs on here is not just 96-well plates, but we also do 2 and 4 mil transfections and 24-well plates. And those are all run in one run on here, and based on the barcodes of the plates, the system will recognize whether they're 24-well samples, 24-well plates with 2 mils or 4 mil samples, or 96-well. We call them 1 mil, but they're a bit under 1 mil in total volume. And based on the barcodes, the system puts the plates on the different liquid handlers to add transfection reagents and cells, and it uses the barcode, a suffix on the barcode, to determine what method to run for that plate. So we treat all the plates as the same and set the system up in an identical manner, and then the system applies different methods based on the actual plate type it's running. So that's a nice feature of the system. And then we recently added a balance to this system, again, for automated liquid handling verification. And then there's a dispenser on here for some reagents that has a nice manifold system. So we have a very viscous solution on on this as part of this workflow. And so it does an automated cleanup and washing of that dispenser after hours. And so that's a nice feature of the system as well. So our DNA gets put on here, we add our transfection reagents and cells, and typically the next day they're getting pulled off of the system and put into Kuhner shakers for roughly a little under a week expression. And so as they come out of that week expression, then we put those plates with cells that have been, CHO cells typically expressing the material over that 6 to 7 day timeline. And then we'll go on to what we call the Beckman quant cherry pick system. We typically refer to this as our IgG system. And so the workhorse instruments on here are the Bionics high-G centrifuges. So these are really nice centrifuges. So if we have different volume ranges and the rotor and the balance tolerance for these centrifuges are very excellent, and we can spin them at very high speeds reliably. And so we decided to upsize the centrifuge on this particular system for that reason. So we have the Bionics high-G centrifuges. We have a fleet of NXs on here. So we pellet the cells in the centrifuge, and then we use the Biomech NXs to transfer supernatant into a fresh plate and take a little aliquot of that into a 384 tilt welt plate compatible with the octet. So we were one of the early folks, obviously with Beckman and in collaboration with Forte Bio, to integrate the octet HTX. And so we take the aliquot of the supernatant, put it on the octet HTX, and use the octet to measure our titers from the crude supernatants. And then that information, using some scripting that was developed with the Beckman folks, we are typically setting a cutoff for what we call an express sample in around 5 micrograms per mil. And then we'll go to one of the other NXs and then cherry pick samples that are above that cutoff, and then leave certain wells or column empty for controls for downstream assays. And so we're typically measuring titer on the octet and then cherry picking and rearraying the samples on another NX on the system. And again, we have a recent balance on here. We have a barcode labeler, so as we make new plates, we're pinging our LIMS system and then coming up with new barcodes and telling LIMS system at the end of the process what these new plates are, where the samples are, and what concentration material they have in them. And so it's a really nice platform that's been really one of the workhorse systems for this and several processes in our discovery efforts. And then I'll talk a little bit later. We recently put some bioshakes on here and where that comes into play. So from this system, we essentially have supernatants of a good amount of expressed material that we're ready to start testing. And so what we have in place, and what we had in place, was these plates that come off the system, and they're variable concentration supernatants, and these values were determined on the octet. And then we would determine what our screening plate setup would be, and we would typically need to normalize these variable concentration samples using a LINX, which is a 96 variable volume pipetting device, to a flat concentration, say 25 micrograms per mil. And then from there, we would go into Tempo software that drives our Echo Access or LabSite Access system and make dose response plates that would be used for screening. And so really the only reason that we're doing this normalization step is because the software that drove this system, it was largely built for the small molecule world and assumes that the plates coming in are all of a uniform concentration. And so we wanted to explore options to go from a variable concentration starting plate to get to the same dose response plate with a software-based solution. And so we work with the folks at Biosero and incorporated their transfer map worklist generating software and the scheduler Green Button Go to drive this system to do exactly this. So we can now take the variable concentration supernatant material, take the same type of dose response plates, and then generate worklists and feed them to the Echo to accomplish this task and eliminate that normalization step. And so this is something that's been in place for a good chunk of this year now, and it's been working nicely. So it's a nice example of a software solution to really streamline our workflow a bit. So once we have our dose response plates ready, then one of our recent systems late last year we affectionately call the Beast, and that's a bit of a play on Beckman assay system. And so it's the drawing doesn't quite do it justice, but the scale is far larger than all of our other systems. To give you a bit of a flavor of the scale, this is a 1.5 meter precise robotic rail on the right, and on the left it's a 2 meter rail. And it's a fully enclosed system with 25 HEPA filters on top, capable of running BSL2 screens. So I'll start kind of in the top left corner and go around the system a bit. So we have two GNF WD2s. They're mirror images of one another, flanked by a stack of Mettler balances, again for liquid handling verification. I'll talk a little bit about that in a bit as well. We have a suite of liquid handlers, including the WD2s, and we have a Certus over a Combi, as well as two Blue Cat blue washers. And then we have, again, the usual suspects, plate locks, A4S sealers, XPeels on here. We have a couple biotech spectrophotometer readers, a multi-mode reader from biotech as well, the Synergy Neo 2, as well as a high throughput flow cytometer, the Z5 from Biorad. And then we go around to a Biomedical i7, and you'll see a couple positive pressure devices off the side of that, and I'll talk about a little bit later on. And the i7 is outfitted with the 96 multi-channel head and a span 8. And then we have the i5 that has a 3D4 head on there. And then a couple cytomats, typically one's always at 4 degrees and one's at 37 degrees. And so it's really, you can see most of the systems I've discussed thus far are built around doing a fairly fixed workflow. This is the kind of jack of all trades where we're going to run everything from ELISAs to cell-based assays to high throughput flow assays. And it's large and it has a lot of equipment, but no screen incorporates all the equipment. You basically are looking at what you want to accomplish with a specific screen and applying the best tools for the job. And we have a lot of tools on this system to be able to accommodate any type of assays our scientists would like to run. And the one thing I want to talk a little bit about is how the balances are incorporated onto this system in particular. And so we have balances that work with our WD2s to have a feedback loop to control dispensing. So when we got this system early on, I put a 2-liter bottle of PBS onto several of the different types of dispensers on the system and just dispensed 20 microliters per well into 3D4 well plates and repeated that for hundreds of cycles. And what you see here, this is normalized to 20 microliters. And the theme of this is basically all the dispensers drift over time in a downward manner. The Certus and the WD2 are using a similar dispense mechanism where you're pressurizing a bottle and opening up a solenoid valve to dispense. And the Combi is a peristaltic pump-driven dispense mechanism. But largely you can see the same type of downward trend over time. This is normalized, and so the scale of this, you can see it's not a massive trend. Over 100 to a couple hundred plates, you might only see a few % difference in volume. But that's something that we can be aware of, and that's something that we can control, actually. And so I'll talk a little bit about that. The other thing is having the integrated balances, it's really an insurance policy. So if a bottle is an ice bucket and it tips over, or if you run out of reagents, you didn't factor in the proper amount of dead volume, we have these systems notify us and stop running and be able to have an operator intervene to fix something and then to continue on. And so this is the balances are a nice feature, a nice addition to the system. And so what we've done with Beckman and other people have done this with other scheduler softwares and integrated dispensers as well, is to have the feedback loop and to have the software control dispense volumes. So in the case of the WD2s, we are able to use that information, feed it back to the dispenser, and as it drifts down, it bumps it up and keeps it dead flat for the course of a run. What I'm showing here is another reagent that we dispensed. This is actually filling up the octet plates that would be on the IgG system with about 32 microliters of buffer. So the octet's typically a 1 to 5 dilution of the supernate, and so we fill 32 microliters of buffer and then 8 microliters of soup. And so here we're filling just a bunch of plates to store in our refrigerator for future runs. And this was early days with this system late last year, and we were sitting there after the system was set up, and I was showing the WD2s to somebody, and they were talking about e-stops and how to control the system and how to respond to errors in the system. And right after it finished dispensing 32 microliters to the 11th plate, I said, well, let's see it. So it finished dispense, and I hit the e-stop right before it released the plate to the robot. And so on the SAMI side of things, you get a pop-up that says, do you want to ignore the error? Do you want to retry it? So just very quickly, the user I was working with hit retry. And so it already had 32 microliters per well in the 384 well plate. And so the retry is going to redo that dispense. And so now you have 64 microliters per well. And so then he was going around like, oh, we need to stop the screen, we need to fix things, and we'll start it back up, et cetera, et cetera. And I pulled him back and like, no, this is the beauty of what we just worked with Beckman to build into the system. Watch the system and just let it go. And so the next plate, obviously it thinks it overshot this dispense. And so the system undershoots the next plate, it overcompensates a bit. And then within a few plates, it dials itself back onto set point. And that's kind of shows the power of this. And so I think he was pretty impressed to see that, and then using this to show other folks of how we're controlling things is a nice example of it. Obviously, this is a gross example, and we typically would not have this much of a discrepancy in control. And oftentimes what people are going to talk about with a feedback mechanism like this, well, how can you catch a minor clog or a single tip when you're using a 8-tip or several channel dispenser? And so we determine the thresholds for what is an error. So usually you are setting your thresholds fairly tight around your set point. And so if you do have 1 in 8 tips clog, you're going to be 12% or so off in that dispense volume. So if you're dispensing 20 microliters, you're going to want to you want to probably keep that pretty tight or within a microliter, so plus or minus a microliter or maybe a couple microliters to be able to catch those types of errors. And so this is something we're pretty happy with and we like quite a bit. And so as we work with our scientists and we get protocols to run different assays, it would typically be something that looks like this. This is a generic protocol of dispensing cells, incubating, doing a transfer, further incubation, dispense, another incubate, and read. So what you typically hand over to your scientists is the reader data that comes off the end of this experiment. What we're able to share with our scientists now is every one of these incubation steps, we have a Gantt chart and we can show timing. And SAMI does that well, as well as several schedulers. But with the incorporated balances onto the system, not only can we show the Gantt chart that we hit all the timing points that they wanted, we can also say, well, you asked for 20 microliters, 10 microliters of antibody, the detection reagent. We can show exactly how much volume based on gravimetric results that these plates were receiving. And so if the data does have certain trends in them or patterns, then we're able to go look at timing as well as dispenser performance to be able to troubleshoot things. So it's been a nice addition to our systems. So this is a recent assay, and so we've done a lot of COVID-19 work over the last few months at Lilly, and a big part of that is running a significant number of ELISAs on this system. And so obviously this system would be very overkill if it were just performing ELISAs, and that's why we have so much equipment on here to do a bit more complex work, cell-based assays and high throughput flow cytometry. But this is something the system has been leveraged for, and it's been performing very well for us over the past several months. And so a typical ELISA process would be, do your wash and block, transfer samples, add your secondary substrate, and then in the system we do several OD reads. And that's one of the nice things is we can do several reads because the plates are always on there. You have a ballpark of, based on our scientists that we're working with, of how long it should take for development time, but since we have all the plates on there and it's an automated fashion, we can read from just a few minutes out to some of the plates we've read at 18 plus hours. And so it's nice to be able to just essentially queue up the system and let it go until these plates saturate, and then we back off and go to the time point or a couple time points before that when we're looking at to try to get good exposure without saturating. You do see a lot of steps in this process for a somewhat basic ELISA. And I would say that that is because of all the way steps. So every way step is a node, and then the script to analyze the way step is another node. And so before and after all these liquid handling steps, you're seeing a balance step followed by a script node. And so this method is significantly larger than you might see on another SAMI-based system. And then one of the nice things we're doing is our samples are coming off in 96-well format, and we're able to use the biomech. These ones are actually being normalized on the links prior to running the ELISAs. So we have a flat concentration of typically up to 96 samples in a 96-well plate. And then we're transferring into 3D4 ELISA plates, and on the I7 we're doing a 4-point dilution. So we're able to run dose response plates in a quadrant format by going from a 96 to a 3D4 well plate. And so we've generated results that look something like this. So the data coming off the system is we're using a DART report to aggregate the data coming off the system, and have made Spotfire templates that really, with minimal touch points, take that DART report and drop it right into a Spotfire template to be able to get what I would call kind of the 10,000-foot view of our systems. And so we're plotting row versus column and trellising by plate. So this is 2 sets of plates, A1 through 8, and then what we're calling B1 through 7. So what I'm typically doing here is sizing by the OD readout. And this is a 3D4 ELISA plate, and what you can see is, let me zoom in here on one of these, you can see a nice titration of some positive controls. So there's controls in the first column, and there are 2 fairly strong positive controls, and you see a nice titration. Again, these are sized by the OD readout. You see towards the end of the A plates and the last plate on the B plates, there aren't any samples in this portion. And so it's nice to see very consistent low sizing of the squares. And then again, I'm looking for streaks, edge effects, other patterns that might jump off the page to indicate a liquid handler issue in the system. And so this is really not the final analysis, but this is what I would throw data into a Spotfire report like this very quickly, or even as screens are running, just to be able to see any trends and make sure that the quality of the data is looking good. And then from this particular experiment, there are some sledgehammer, like really, really strong hits in plate A2 that ended up becoming important for us later on. And so from this system, I'm really trying to hit home the point that the functional highlights on here, we can run ELISAs, very wide variety and flavors of ELISAs, cell-based assays, high throughput flow, and then we can even prep plates for offline instruments. We have offline high content imagers, so we can do all the washing, fixing, staining of the plates on here and seal them up and prep them for a high content imaging based assay. And so it's been a nice, a really nice system. Again, kind of jack of all trades on here, and we've got a lot of equipment, and we've really been focusing on ELISA the last few months, and so we're looking forward to returning to work soon and hopefully getting more cell-based assays. And we, late last year and early this year, we got a little bit of high throughput flow, some high throughput flow cell binding screens under our belts, and so we're excited to continue on that effort when we can get back to some normal projects. And so I touched on this whole process and going from single B cells to screening results to generate lead molecules. What I mentioned a little bit earlier when we were talking about the transfection system is, this is all in 96-well format, and the expression system, we're doing 1 mil samples. As we generate these leads and triage that list down, the samples do feed back in here a bit, and we decrease the number of samples, but increase the volume and the scale of those samples. And so we typically go into 24-well plates up to 4 mil cultures. And at that point in time, we're typically not working with crude supernatants when we're going for our testing anymore. We'd like to purify that material. So early on, last year, year and a half ago, we were scoping out a DNA purification system as well as a protein purification system. I think you can see here, we purify a lot of DNA. When we get to the protein purification side of things, we've down selected the number of samples pretty significantly. So it took a step back and tried to justify that we should spend the money to upsizing the DNA system and then do some small modifications on our existing systems to enable the protein purification side of things. And so that's where I'm going to talk about those little tweaks that we've made recently, and how we can enable the throughput that we need for protein purification. And I've already shown why and how we've upsized the DNA purification system. So kind of reallocating the money where we get the best bang for our buck. And so this is the Beckman Quant and cherry pick system, again, what we call the IgG system. And so as we pellet cells and harvest the supernatants and we measure the titer on the octet, well, we can run 24 well plates in the system in largely the same manner. But what we can also do is use the fleet of liquid handlers on here to add resin to those supernatants. And then we seal the plates up and then put them on the bioshakes for roughly 30 minutes or so. Then what we would do is pellet those beads of resin in the high-G centrifuge. And then where you typically are having roughly 4 mils of sample and resin in the plates at that point in time, take off you've pelleted the resin, so it's in a pellet at the bottom of the well, take off the roughly 3 mils of supernatant remaining on top of that. So what you have as an output of this system is around 1 mil of liquid and resin in these 24 well plates, and the antibody is largely bound to the resin at that point in time. And we've done it. Mike Cohen's done a nice experiment showing that we are capturing nearly all of the antibody in the supernatant. And so from here, we can go to the DNA purification system where we have the new i7 with a couple positive pressure devices and use the i7 to transfer those roughly 1 mil of sample and the resin into 96 well filter plates, and then use the positive pressure devices to push the sample through the filter plates, do washing, elution, and then use the Biomech to make an aliquot. Again, read it on the integrated spectrophotometer, and then have the i7 take a work list in and break out those samples by scientist or requester or user. So from here, we will measure the amount of material and then break the plates out and give people plate maps of their purified material. And so again, putting a few bioshakes on another system and a couple positive pressure devices on this guy, and using all the other equipment that we already have in place, really is a little bit of equipment to enable a whole nother automated process that we weren't doing previously. And so again, this is the overview of the process. And as I talked about in the very beginning, a lot of this is tied together with a LIMS infrastructure, and a lot of folks have been helping on that front. So it's a huge effort, not only to build these systems with our team and the collaborators such as Beckman and Fujifilm and Biocero. It's been a lot of work with the informatics folks and the IT folks to develop the infrastructure, the LIMS infrastructure for these. And so from this whole process, and the whole reason why this all exists is to really deliver multi-species ELISA data, cell binding flow data, functional cell assay data. And then when we purify DNA back on the DNA purification system, we do make a cut for sequencing as well. And so by the time you have your screening data coming off the right side of this process, you have sequencing results as well to tie that back to. So it's really a rich data set to give our scientists as much information as possible to whittle down our lead molecules. And so with that, again, it's a really huge effort. I joined a couple of years ago, so I'm very thankful to have the pleasure of sharing the work from the group. But this, Tom, Melissa, and Mike had a huge role in this for several years before I got here. And David, Liliana, and Andrew are in my group and help with the automation side of things as well. And so everyone in my group is Mike, David, Liliana, Andrew are responsible to support and keep these systems running and work with Beckman and the other vendors to improve these systems and evolve them over time. And our scientists that carry out the single cell process, our screening group, as well as our sequencing folks, it's a huge amount of people in what we call the automation group, all underneath Tom, that use these systems, have suggestions, help evolve these systems and improve them. And then the scientists we work with upstream and downstream of us, as well as the IT folks, have a huge role in this. And then we have several labs managers that help support what we do. But we're very thankful for it, and the management support to be able to build out this infrastructure. And again, the people we're working with at Beckman, Biocero, and Fujifilm to help develop these systems and to continue to improve them over time, incorporating balances, integrating new devices as they come out, exploring new technologies and workflows. We have a lot of stuff to work on as we continue our process and look for new opportunities to incorporate new technologies, whether it be biology-driven or automation-driven. So it's always it's a never-ending cycle of improving these guys.
Well, I want to thank you, Paul, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box on the far left of your screen, and we'll answer as many questions as we have time for. So let's take a look. We already have some incoming questions from our audience. Our first question, Paul, do you have a general sense of hands-on time required to set up and kick off a runs in each respective module? A follow-up would be, which process has offered the greatest ROI for automating?
Yeah, thanks. Can you hear me okay?
Yes.
Oh, great. So there's quite a bit of setup for several of these systems. The DNA purification system in particular is one that's quite involved due to the fairly large number of labware that are required for runs. So you have systems that can take anywhere from 10 to 15 minutes to that system takes upwards of a half hour, possibly a little bit more to set up a system. We've been trying to ease some of that a bit from our scientists. For the Skylines, for example, we're having some lab support staff that are able to preload the Skylines with the QIAGEN 96 kit filter plates as well as tips. So we have extra racks for the Skylines that we'll keep preloaded on the side of the system. So when our scientists need to run the system, they can come over and pop those on rather than unpackaging all the consumables and loading the towers themselves. Additionally, we've put in some cleaning and some priming and cleaning systems on that automated mini prep system, the DNA purification system, as well as our transfection system. We've put in those cleaning systems that allow a user to really plug in a bottle, and they don't necessarily need to then prime the system or clean it at the end of the run. The system will go through a series of switch valves to prime with the reagent of interest at the beginning of the run. And at the end of the runs, we have scripts then to flip those valves to cleaning reagents to then decontaminate and clean the dispensers. So that's also helped in some of the setup time. We are looking at other things too, future technologies. One of the things we're thinking about in the mobile robot space is using those to preload these systems in the middle of the night. So you could think of this fleet of islands of automation that we have. We could have racks of tips, plates, other types of labware, where then a mobile robot, you would tell however many plates you want to process on each of these systems, and it would pre-fill those systems with those tip boxes and those plates ahead of time. And then the user in the morning would come bring their sample plates on and fill up any reagents or troughs. So that's something we're thinking about. We haven't moved forward on it yet, but one of our ideas.
Thank you so much, Paul. Now, has the system been used to generate other biotherapeutics such as peptides or other recombinant proteins?
Yeah, that's a good question. So we kind of have this islands of automation approach where you don't have to start at A and end at Z through this process. When we have antibodies, and that's our kind of workhorse platform, those would typically go through all the platforms that I discussed here. We have a lot of work in other types of therapeutics, such as peptides and other types of VHHs and whatnot. So we can jump onto these systems at different points. So if we have someone doing the molecular biology and the cloning in a different setup, we can jump onto it at the next phase of the process. We will have concessions on some of the systems. So when we go express different things and show if they're not antibodies and there are other types of samples, when we go to harvest those samples and measure how much we have on the octet, for antibodies, we're using the protein G biosensors, but for other types of samples, we use Pro-A biosensors, or we use the streptavidin sensors, and we code on their different antigens. So we have some flexibility across our platforms to be able to adapt to the different types of biology and the different types of samples people want to process through our systems.
Thank you so much, Paul. And I want to thank our audience for these great questions that are coming in. Our next question, are you doing any further work on protein purification, such as DOE for resin selection, meaning the upstream process development on the system?
Yeah, so on the systems, primarily, we're automating fixed process and pretty well-established processes. We do have some flexibility in there, like I just mentioned, for the different types of samples and how we handle those on some of the systems. For optimizing protein purifications, as an example, we typically do that offline, using a lot of the same tools, ideally, and the same instruments, but we work out those details off these systems. And as we're converting from, say, MABS Lecture to Prism A, or different types of resins that we're evaluating, that would be done in a more manual offline manner in those matrix experiments. And then once we've worked out those conditions, then we update our automated systems, our automated processes with those new conditions. And so we've had to make adjustments. We have some upgrades coming in the early part of next year due to some of those changes where we're tweaking our purification, and we need to do a little bit different type of elution in a larger volume. So our elution plate is going to be not a normal standard height plate. It's going to be a much taller plate. So we need to look at different types of devices to be able to accommodate a little bit taller stack of labware. So that's something that usually happens offline, and then we really try to automate something that's a little more solidified. But we always try to build in the flexibility that's necessary. If our workhorse protein purification platform is pretty well established, and we're really trying to automate the lion's share of what we're doing. When we have his-tag proteins and other types of proteins that we do need to purify, those are much further and fewer between. So we're most likely going to push those to more manual purifications because they're not very frequent, and focus on automating the lion's share of the work and kind of really relieving the major bottlenecks.
Thank you so much, Paul. And we have time for one more question. Have you considered automating any cell-free protein expression work on these platforms?
It's a topic that's come up from time to time, and I would say we're a service part of the organization. So if our biology and our scientists find the need for that, and we have projects that need to go down that route, then we'll find a way to help with automation. Like I mentioned earlier, we could automate different aspects of it. It could be kind of A to Z process, or we could automate different components that would fit these different types of workflows, and try to just relieve bottlenecks in that manner. So I would say we don't have cell-free expression platforms automated at this point in time, but that's something that could be changing. The molecular biology system is another example I'd highlight, where the type of mobile and the reagents and the process that we do on there today are very different than what we were doing 3, 4, 5 years ago. So not only does our hardware and software and automation components of the systems change over time, but the biology and the processes significantly change over time as new kits become available, new standards of, like, best practices, and new reagents become available. We adapt our process to try to take advantage of the latest, greatest technologies in all respects.
Paul, I want to thank you for your time today and for your important research. Would you like to provide any closing remarks to our audience before we go?
I just want to thank Beckman for sponsoring this, and LabRoots for hosting the webinar, and I look forward to connecting with more people on different platforms in the automation space through platforms like this and the conferences coming up early next year.
Well, I want to thank you again, Paul, for this great presentation. And I also want to take the time to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and bye-bye.